Morning, everybody. Today, the lecture topic is enzymes, enzyme kinetics, and enzymes as targets for drugs. Let us see the learning outcomes first. Understand why enzymes are the most important catalyst in biological systems. Understand the basic enzyme structure and its components. The third one is state what a cofactor is. Distinguish between coenzymes and prosthetic groups. State why cofactors are needed for certain enzyme reactions, and then explain the models of enzyme substrate binding. Explain how enzymes lower the energy of activation and increase the rate of reaction. Discuss the factors affecting the rate of enzyme catalyzed reactions. Then describe the Michaelis Menten question and use the equation to predict the Km and Vmax. Discuss the significance of Vmax and Km. State the major classes of enzyme inhibitors and predict the mechanism of binding and impact on Km and Vmax. Let us start with the introduction. Almost all reactions in the body are mediated by enzymes. The cells cannot be seen by the naked eye. In these cells, there are millions of biochemical reactions are taking place. These reactions are catalyzed by certain molecules. These are called protein catalysts. They expedite the reactions without being consumed in the process. And majority of these enzymes are proteins. Notable exceptions are there. These are RNA molecules and called as ribozymes. And these enzymes have certain general characteristics. Mostly they are globular proteins. As you all know, the proteins can be divided into fibrous proteins, globular proteins, and structural proteins, functional proteins. The enzymes are globular protein, but not always. They increase the rate of reaction without being used up. This is one of the typical characteristics of a catalyst. They never used up in the reaction. And their presence doesn't affect the nature or properties of end products. What that means, basically, the enzymes doesn't react with the product and change the product characteristic and nature. They need in small amounts to accelerate the biochemical reactions. Since they are not involved in the reaction, only involved in catalysis of the reactions, these enzymes can be recycled. That is the reason small amounts are used to accelerate the biochemical reactions. They are very specific in their action. This is what called enzyme specificity. When you say specificity, it can be a substrate specificity, or it can be a bond specificity, it can be an isomer specificity. Example, sucrose catalyzed by sucrase, maltose catalyzed by maltase. So maltase is a specific enzyme for maltose. Glucose oxidized by glucose oxidase. So glucose oxidase is a specific enzyme to oxidize glucose. And they lower the activation energy of the reaction. The reaction which doesn't need the activation energy are spontaneous reaction. But certain reactions need to be activated by giving an energy called activation energy. Enzymes lower this activation energy. And some enzymes require cofactors for their proper functioning. They are sensitive to pH, temperature, since they are proteins. Let us look at the structure of enzyme. The protein component of the enzyme is generally referred as apoenzyme. It is larger molecular weight component of the enzyme and contains the active site. Some enzyme needs cofactors, some doesn't need cofactors. Enzymes need cofactors. These cofactors attach to the enzyme or apoenzyme. Apoenzyme and cofactors together referred as coenzyme. What are these cofactors? Cofactors can be either small molecular weight or inorganic ions. When the cofactors are organic molecule, they are referred as coenzymes. Coenzymes are the complex non-protein organic molecules that participate in catalysis. Some can be synthesized in the body. Others contain a vitamin or vitamin derivative involved in specific reaction type. Again, these coenzymes can be divided into co-substrate and prosthetic group. What are co-substrate? What are prosthetic group? Co-substrate associate with the enzyme only for the reaction or at the time of the reaction. And prosthetic group generally monogamous and permanently bonded to the active site, covalently or non-covalently. These co-substrate can be chemically modified in the reaction. Some of the examples for coenzyme as co-substrate: adenosine triphosphate, ATP, guanosine triphosphate, GTP, NAD and NADP, coenzyme A, S-adenosyl methionine, tetrahydrofolate. These are some of the examples for coenzymes as co-substrate. Examples for coenzyme as prosthetic groups: flavin adenine dinucleotide, heme, biotin, pyridoxal phosphate, thiamine pyrophosphate. When you look at overall, cofactors can be divided into coenzyme and metallic ion. Coenzymes further divided into co-substrate and prosthetic group. All coenzymes are cofactors, but all cofactors are not coenzyme, because cofactors can be either metallic ion or coenzymes. What is the role of cofactors in the enzyme function? Suppose we assume there are two substrates going to bind to the enzyme as indicated in the picture. As the substrate approach the enzyme, they cannot bind it easily. The reason, the active site of the enzyme or the binding site of the enzyme is not complementary to the substrates. In such case, cofactor modifies or changes the conformation of the active site, which allows the substrate to bind easily. So the first role of cofactor in enzyme action, cofactors changes the conformation of active site. The second function, cofactors participate in temporary bonding between active site and substrate. In the presence of cofactor only, the substrate binding is complete. So there are two functions of cofactor. One is cofactor changes conformation of active site. The second one, cofactor participates in temporary bonding between active site and substrates. Some of the inorganic elements that serve as cofactor for enzyme. As you see in the slide, Cu2+, Fe2+, K+, magnesium, manganese, molybdenum, nickel, selenium, zinc can act as cofactors for various enzymes as indicated in the PowerPoint. As I said earlier, enzymes lower reactions activation energy. Let us see what is activation energy. Let us see this picture. Lactose converted to glucose and galactose. This is a hydrolysis reaction. In the absence of reaction, in the absence of an enzyme, the conversion of lactose to glucose and galactose need an activation energy. As it indicated nicely in this picture, you can see we imagine the lactose as the boulder. The boulder has to be pushed uphill on the mountain or hill top and then get activated after that, the reaction takes place, and you get glucose and galactose as a product, and then energy will be released, splitting the lactose. So the energy we use to push the lactose uphill to the top of the mountain is, we call it as activation energy. Lactose had to undergo that barrier, energy barrier, to give glucose and galactose. What happens in the presence of enzyme? In the presence of enzyme, the activation energy is reduced. The enzyme substrate complex forms a different kind of a reaction, and that allows the lowering of activation energy, and which leads or which facilitate the enzyme catalyzed reaction taking place very fast. But the net energy released is always constant. So you can see this as again indicated. When a substrate is going to be converted to product, you get an energy barrier. What is happening now? In the presence of enzyme, the energy barrier is reduced. In other words, the activation energy is reduced, and then same product is formed. This mechanism by which enzyme enhances the rate of reaction. The first, by providing an alternative energy favorable reaction. All chemical reactions have an energy barrier, which is called free energy of activation. Usually, there is an high energy intermediate transition state. Then active site chemically facilitate the reaction. When the substrate binds to the enzyme, stabilizes the transition state. In this process, there may be gain or loss of electrons, may form covalent links too. Let us see this picture. Suppose when you have a carbohydrate meal, the complex carbohydrate digested and converted to glucose. Glucose absorbed in the intestine and enter the portal vein and enter the cell, enter the liver parenchymal cell. So when a glucose molecule enter the cell, glucose is immediately phosphorylated to glucose 6-phosphate. Can anyone tell why glucose is converted to glucose 6-phosphate without being converted? Glucose cannot be metabolite, is it? What really happens? Usually the glucose is a neutral molecule, isn't it? When the glucose enter the cell, because it's a neutral molecule, it can easily escape the cell. So what we do is, when glucose combined with ATP, right, a negative phosphate group is added to the glucose molecule. Since the glucose 6-phosphate is a negatively charged molecule, it cannot go through the hydrophobic lipid bilayer of the cell membrane. So let us see this reaction. Glucose plus ATP, when they act as reactants, the product is glucose 6-phosphate and ADP. In this graph, you can see glucose plus ATP, when they react, right, in the absence of an enzyme catalyzed reaction or uncatalyzed reaction, what happens? The glucose plus ATP form a transition state. The transition state, to reach the transition state, we have to give an energy. That energy is called EA, or activation energy of the uncatalyzed reaction. Suppose if the same reaction is catalyzed by an enzyme, usually the enzyme convert the glucose to glucose 6-phosphate are hexokinase and glucokinase. So in the presence of the glucokinase or hexokinase, as you can see, the activation energy is reduced. So enzyme act in a biochemical reaction by reducing activation energy of the reaction, and overall the reaction, the the energy status of the product is not affected. Only the activation energy is reduced. Let us see how enzyme binds to the enzyme and substrate bind together and form the enzyme substrate complex. In each enzyme, there is an active site in which the substrate goes and bind and the catalytic reaction is taking place. When the substrate bind to the active site, there is the formation of enzyme substrate complex. Then the enzyme substrate complex become enzyme and product. The enzyme can be recycled again and again to continue the reaction further. So when you look at the enzyme structure, the active site consists of a site called catalytic site that is indicated by this dark blue region. At the same time, the cofactor also attached to the active site. When the substrate